But the reason I'm here is because of the invitation from the Metcalf group to come speak with the journalists, because as a scientist, I'm really committed to getting this kind of information out to everyone so that everyone can understand it. I'm also really committed to the idea that actually my research in some cases is funded by the federal government. So I am your tax dollars at work, and therefore I should be asked to come talk to you about what we're doing. So with that said, I'm really delighted to be here, and I'm really looking forward to talking to everybody when the talk is over. So what I wanted to do today is 4 different things, and we'll see if I succeed. I want to sort of get us a little bit of ocean acidification 101 so we all understand it a little bit, and maybe for the fellows, an extra little spicy bit of yummy chemistry to share with you, because I know you're going to do that later. I want to talk about whether organisms can adapt to the coming changes, because that's really one of the biggest questions we face right now. And I want to do 2 things at the end. Is I want to share with you what we're learning as a research community, what we think about how we view adaptation, and whether all species are going to be wiped out or whether some can make it. I want to share and kind of translate some of the science and share with you what I'm hearing right now from my colleagues. And lastly, I really want to end with what can we do right now, because my message is going to be that we've learned a lot and there's a lot of local action that I think we can take as coastal communities that makes an enormous amount of sense if we act together as communities of scientists and journalists and citizens and other agencies working together. So at the end, I think you're going to have to live through some of this science stuff and maybe some news that makes you not feel so great. But when you get to the end and you see beautiful seagrass beds and beautiful kelp forests, then you know the good news is coming and the talk is almost over. And beverages and snacks await, right? Because I know that I'm the only thing standing between you and a really nice reception. So let's get this rolling. So as promised, let's talk about ocean acidification. So these slides, the next series you're going to see, were shared with me for this presentation by Richard Feely, who's the lead scientist at NOAA at the Pacific Marine Environmental Labs, and he's one of the world's experts on this topic. So Richard asked me to share this with you. So ocean acidification really is the change in the pH of the surface oceans because carbon dioxide is absorbing into that ocean. And what happens is when that happens, there's some chemistry that occurs such that the pH is lowered. And this has consequences for marine organisms and marine ecosystems, and that's where we're headed in the part of this talk that's not going to make you feel great, right? So it's pretty clear, many agencies and many research groups have been looking at this, that we have many sources of anthropogenic CO2 emissions to the atmosphere. It couldn't be any more topical to try to talk about this today, given the action of President Obama yesterday and the news that we were going to try to take action on some of our emissions in the United States. Certainly coal-fired power plants and vehicles are our 2 greatest sources of carbon dioxide. And one of the things we know is that anthropogenic CO2 goes somewhere, and a scientist would call them sinks, places they go. And if you just look at the bottom one, we're going to cut to the chase here. Oceans. Oceans, we estimate, have absorbed somewhere between 27 and 30% of the carbon we've emitted, since the Industrial Revolution. And lest we make fun of this, this is a really important point. So a cartoonist kind of captured some of this, where scientists were thrilled when they realized that the ocean absorbs greenhouse gas CO2, because it was a service that the ocean was doing, that it was literally removing pollution from our atmosphere, where we've been using the skies as kind of like a sewer, really. And even so much so that in the 70s and 80s, people were talking about injecting carbon dioxide into the deep oceans to store it. And what happened in about 1999 to 2000, especially coral reef biologists came forward and said, wow, this is going to change the pH of the ocean, and it's going to change the way organisms who make hard parts, who make shells and skeletons, things we eat, oysters, coral reefs, things that other countries rely on for almost their entire economy. And that's when everybody went, oh, and you can see the science boat going, oh no, this really isn't a service anymore. And so what happened then was, over probably the last 10 to 15 years even, there's been an enormous effort internationally and within this country to ask, how are calcifying organisms impacted by changing chemistry? And so we went through this eruption, literally, of research in this area to try to understand these things. Now, this is the point where most people who are deeply involved in the science really want to know, yeah, well, great. Is the ocean really changing? Is its pH really changing? And the truth of the matter is we don't have very many places on the planet where we have long-term data about this, but we do have some spots. So let's just talk about that for a moment, okay? So oceanographers have a few sites around the planet, both in Bermuda, and what I'm going to show you is the Hawaii Ocean Time Series, HOT we call it, and they call it Station Aloha. It's kind of a cool name. It's a buoy out in the water, and it's got instruments on it, and people have been recording pH and dissolved carbon or CO2 levels there for quite some time. And what you can see is the commensurate increase over time of CO2 and also the decrease in pH. And this is one of our best long-term study data sets that we have, and we don't have very many of them. And really, it's the oceanographic community at places like Rhode Island. There's been participation from many groups at these sites collecting these data. By the end of this talk, you're going to realize that one of our real shortfalls is whereas we know some things about the open ocean, which is great, and that can tell us the trajectory we're on, we know very little about what's going on the coasts, the place where a lot of our food comes from. So this is something that I think we can check a box and we know what to do next. I think that's a really good point. So if this is what's going on with CO2 dissolving into the water and the pH is changing, how do we get there? So this is the hairy, scary chemistry slide, right? So for the fellows, right? You're like, no. So what happens is the CO2, it's chemistry. So CO2 in the atmosphere dissolves in the surface of water. And this is where, as a journalist, it's not climate change, because that's weather. This is just global chemistry. So if you took a glass of water that had a pH indicator and you went, you change the pH of that glass of water because your exhalation has a bunch of CO2 in it. And it's that simple. So it's a little scary to say this, but this kind of reaction is inevitable. And so what happens when the action happens is there's a dissociation of carbonic acid into a proton and bicarbonate. And it's that proton that makes the pH change. The more protons, the lower the pH. And furthermore, there's additional bad news though, because what happens is eventually, a carbonate ion is scavenged to make bicarbonate, okay? So the bad news, pH declines and carbonate ions are scavenged. Why is that bad? Well, this is the part that the coral biologists knew that nobody else knew to pay attention to until the chemists and the biologists got together. Because over time, what's going on is carbonate ions are the building blocks for those hard parts, for those shells in the coral. And there's a projection now that as the ocean acidifies, so too does the carbonate ion concentration. So you can think of it as building blocks in the water going down. And it is a little bit complicated, so don't look at this, but this is again for the journalists. Nobody else look! So what's going to happen is when you do your chemistry, there'll be this component of asking, well, there's this happy balancing place in the water, the saturation state of how calcium and carbonate ions come together, and this animal is collecting those and making a shell. My analogy, and it's not a great one, but it's like making rock candy. You ever done that, where you put a string in saturated sugar, and you've got to have enough sugar in there for the crystals to form? It's kind of that same idea. These organisms need to have enough of these things in the water to form their shell. And ocean acidification reduces the availability of that material. So that's the chemistry. The biology community went, ooh, that is bad. And so this is a little dated now, but you can see this figure showing the year on the bottom and the number of papers. There was this massive uptick. It only goes to 2011, but that number will keep going, where people, almost out of a sense of service to the oceans, retooled their research programs to look at this. And there's a professor at URI, Brad Seibel, who was one of the leaders in this field, who said, well, I'm going to work on this. And a bunch of other people did as well. And over time, we can do a thing called meta-analyses. There's a scientist, Christy Croker, who scraped together everybody's data and said, let's look at the accumulated research up until about 2011, 2012, and see what we can find. So this figure shows calcification. So that's the process where everybody makes their shell. And the dashed line is no effect, and anything below it is deleterious. Everything above it is just barely okay or breaking even. And so the dominant thing here is you can see bad news for many organisms in calcification. This figure shows growth. Here's Christie summarizing all these studies on growth. This is where you start to get bummed out, like, but you can see that things that photosynthesize, diatoms and algae, seem to not be impacted by this. And this is where the winners and the losers discussion came from, that maybe photosynthetic organisms are going to do well because of all the extra CO2 in the water. And we'll touch upon that topic later. Another interesting thing that Christie found was that when people look at organisms, especially those that are food items like oysters and mussels, that there was a difference in life history stage vulnerability. So that early stages, at least in the meta-analysis, look to be a little more vulnerable. And these results have been obviously really noticed by mariculture folks. And in California, our sea urchin divers are concerned that the health of sea urchin larvae and their settlement to become adults is threatened, because in our neck of the woods, we don't have lobsters, we have big red sea urchins where the gonads inside are the uni, the sushi uni, worth a lot of money to our neck of the woods. So this is where we are right now. And about 2 years ago, we started to realize a couple different things, and that was that certainly evolution works. Organisms must have tools to respond to these changes in the environment. And thus began an exploration led by many people from around the world who are actually evolutionary biologists who got involved in ocean sciences to address this question. And this is going on all over the place. Just as a sidebar, in conservation biology right now, there's a paper that came out in Science. The lead author is Dawson. And in this paper, this individual defines the word adaptive capacity. And they call it a balance between physiological tolerances and the ability to adapt through true evolutionary change. And what you would say is, wow, well, it's really warming up, and these butterflies are really important pollinators for these local plants. What's their adaptive capacity in the face of a drought? And what that means is, what tools do they have, all of them, to face these changes? So this is a real movement in conservation biology, and I'd recommend that the journalist folks read this paper. It's very accessible, and it's got some good ideas in it, and a lot of people are referencing it now. But let's come back to that adaptation idea in the oceans. Can organisms actually change their genes, change the way they are to respond to future changes? And this is, a bad pun, but it's a sea change in ocean acidification research, because something happened that changed the way we thought about this problem. When the whole issue started, we had that open ocean data that showed that pH decline line, and we were all like, oh no, everything is going. And when I got involved in it, I work in coast, and I thought, well, I'm just going to go out and get a little pH-ometer to record pH to see what's going on in the environment. And it turns out there isn't anything like that. So I had to rely on the incredibly handsome Todd Mertz from Scripps Institution of Oceanography. He's an ocean engineer. And together with Ken Johnson from the Monterey Bay Aquarium Research Institute, they literally MacGyvered a sensor for the community, where they took a glass electrode made by Honeywell and they put it in a tube. Here's one of our sensors deployed in the Santa Barbara Channel, and they put in a memory device and some D-cell batteries, nothing fancy. And we had a programmable sensor that could record pH for us continuously. And what this meant was we didn't have to be the big old rich, scary oceanographers, and because we didn't have the kinds of resources on the coast that we need to deploy those types of sensors. And the really important point here is that after Todd got these out to us, we helped beta test them along with other biologists and ecologists in many different locations around the world and many within California. So along the bottom, you can see from left to right a trace from a coral reef, pH recorded on a coral reef. In the center there, pH recorded in the coastal waters of Antarctica, and then over here, my local kelp forest. And the first thing that pops out at you is, wow, no ecosystem is the same. Some of them have really rhythmic variability, like that daily change in the coral reef. That's actually the respiration of all the symbiodinium algae that are these symbionts inside of corals. They're so numerous that they change the pH just with their own metabolism. And over here, it's crazy town. And what we've got going on in kelp forests is that there are these huge macrophytes, big kelp, other algae that are so active in respiration that they change the pH that much. So we're going to come back to this idea. But the thing that you want to look at is the variability. And if you're an environmental biologist and you see that suggests that adaptation should exist. You think, oh, there's selection going on. There's some genotypes out there in the California coast that are used to this low pH every given Tuesday. Because if you go back to these data, we were a little surprised to see this, but maybe not really. But you know, the IPCC was projecting disaster at levels that we saw in right here, these low levels, that by the year 2100, that would be the pH of the open ocean. But we're seeing it right on our coast because of biological processes out there. So organisms are used to this, to use a different term. And what we want to know is, how flexible are they? How much plasticity do they have? And can they adapt? And a lot of people in the OA research world thought, yeah, actually, they probably can. And so I want to share with you just really briefly why we think that is as a group of biologists, and why a lot of people are focusing on the adaptation question, and furthermore, why it's so important, right? So we'll just kind of talk about that. So first, one of the things that people thought was, well, evolution takes a really long time, and we don't have that kind of time because ocean pH might be changing really fast, and climate change is a rapid process, we believe, and we know. And so no one thought that maybe those two processes could keep up with each other. But if you go into the scientific literature and you read, there are people that are studying rapid evolution. So there's a guy, David Resnick at UC Riverside, who studies guppies. And he's shown that these guppies in Trinidad can evolve really quickly, something that we wouldn't have expected from something that wasn't a bacteria, for example, right? So we have evidence in the literature that this is not biologically impossible. There's some laboratory experiments that if you look at them, this is an experiment on yeast, where yeast are exposed to high salinity, let's say. And then you see this, whoa, death spiral in the cultures. But after a while, the resistant genotypes take over and they start to multiply and the culture comes back. So these timescales are totally different, but evolutionary biologists know that this is going on. And the way we think about it is that if rapid evolution or evolution is going to occur, there has to be some genetic variation in the population, some grist for the evolutionary mill. So right away, it's really important for fisheries biologists to start asking, whoa, how much genetic variation is in my salmon population? And that's what a lot of people in Alaska are doing right now, obviously looking more maybe at warming in river drainages than in acidification, but that's a really important component. And now, the other grist for evolution is new mutations, and those are fairly rare. So you'd sit around waiting for a long time for that to happen. And then in many species, it's not possible to study it, right? But for OA studies, a lot of people now have been doing some really simple things to explore, to look for this genetic variation that tells us about the plasticity and ability to adapt. And so I want to share with you some of that science, because this is the really important stuff that came in the last 2 or 3 years that fully gives us hope, right? Because I think in about 2010, it was like, oh my goodness, this is really, really bad. And now by bringing more investigators into this field and looking at things with a different kind of lens, I think we're learning a lot. So let's just 3 or 4 slides to talk about that. So firstly, we're learning about variation between genotypes and stocks and strains. So some oysters have different genotypes and they're better at resisting these phenomena. There are clonal differences in some organisms. So we're really seeing that organisms do have genetics to respond and to resist their calcification being shut down by ocean acidification. The other thing that we're learning is that this thing called local adaptation, where organisms become fine-tuned to the acidification or pH environment that they live in. It's like getting used to your local neighborhood. And so I want to talk to you about this, lest we go down a rabbit hole, but it connects to the importance of location, and actually on a small scale, right? So let's talk about this a little bit more. So the question we're asking is there a local adaptation to gradients out there in the environment? Let's go look for places where pH changes and see what the animals are doing. And this is a research project that I've been working on for a couple of years, so I'll show you some of the data. But the reason we're interested in it is because the other way to turn this argument around is that if you find places where there's a lot of variability in the environment, the likelihood of having resistant genotypes there is high. So those places where it's already crappy, right, like low pH, there's already selection going on. And we would almost argue, especially in the California marine protected areas right now, that if we find those places, they ought to be protected as hotspots for adaptation. And similarly, you could find places that don't have very much variation, and they might be protected as refuges from future acidification, the place where pH change will hit you last. So ironically, we find ourselves wanting to protect places that are already really variable and crazy. So the management strategies here are becoming quite interesting. And the conversations I'm having now, I never would have had 5, 6, 7, 8 years ago. So let me just show you, we're going to drill down into the science now, so it might get a little bit boring, but hopefully not too bad, right? So on the US West Coast, we have this process called upwelling, where wind blows over the ocean and this deep, cold, acidic water comes up onto the shore. And we're finding that it comes all the way up on the shore into the intertidal and bathes all the organisms on the coast. And we learned this by putting out sensors. We unified across the West Coast from Oregon down into California, and we showed, where's my ginormous pointer? Yeah. And, ooh, it's like a fishing pole. So we showed that if you look at the latitude, so here's Oregon and here's California, and here's our scale. Red is low pH, the heat map, and blue is alkaline. What you can see is that down here in California, there's much less of the low pH events. And even in Northern California, it starts to have more low pH, intense upwelling signal. By the time you get to Oregon, there's a lot of it. So the hypothesis is that northern populations should be tougher than southern populations in the face of OA, because those have been hotspots for adaptation. So what we did was use one of our relatives to the fisheries species, this little thing called the purple sea urchin. And it's really a great choice because it's found along the whole coast, so we can compare populations and look for the genetics. And they build a calcium carbonate skeleton. And in this case, we actually use the babies, the larvae. And just for a moment, the reason we do that is they're really easy to raise, tons and tons of them in culture. So you get lots of genotypes in a bucket. And they also, when they make their little calcium carbonate skeleton, it starts as an early embryo, like just a tiny crystal. And then it turns into this jack-like looking thing, a triradite spicule. And then as the embryo grows, it becomes something like the poles in a backpacking freestanding tent, so that the skeletal rods in the embryo are the poles and the tissue's like the tent draped over it. And the little thing swims through the water and eats and eventually settles out to become an adult. But you can work with these in the lab and they're perfect because there's so many of them. And so what we do is we raise them, we mimic future oceans in buckets in the lab, spawn urchins, do fertilization and put them in there, and then we measure the size of their bodies after they've grown for a little bit under different treatments. And so we just can measure thousands of these things because you get hundreds of thousands of eggs and sperm from one male or one female. And you can do really neat crosses in the lab. And basically what we found after just taking urchins from all over the place we find that when they grow under low pH conditions or high CO2 that mimics upwelling, they're smaller. So their body size shrinks, probably because the energetic cost of making that skeleton is high, is what we hypothesize. They don't die. They make it. They're just littler. But that's what you graph if you're like a physiologist. You're just lumping everybody together, right? But if you're a population geneticist or somebody interested in genetics, you might graph it like that and show how the different families, the different pairings of male versus female, how their sizes are different. So what we discovered was that there was genetic differences across the different crosses, where some of the progeny were better at resisting pH and retain their body size, and some of them were not as good at it. And if we did some population genetics that I won't talk to you about, this is kind of what we finally determined. This is a little weird to look at, but just look at that red arrow. It matters who your daddy is. Because if the sire of a cross was from Northern California, the progeny could resist low pH. And this doesn't seem like that big of a deal to you, but when you do the population biology on it, it's pretty massively important, because that trait we found was really, really, really heritable. And it suggests that out in nature, there are all these traits that could possibly respond to low pH or warming. And so that natural populations may already contain some of the genetic variation to withstand climate change, and that we just need to look for it and possibly choose the species that we want to look at. Is it a fish? Is it lobster? Is it oysters? So there are targeted studies that we should be doing. The other thing that we're learning while we do this, if genetics matters, is that there are carryover effects. So this is going to be the last data thing that will eventually bridge us to the good news. So one of the things that we learned while the whole community was doing these early life history stage studies, remember I said early stages, embryos, and little juvie things are more sensitive. This is a study by Annalise Hettinger, who looked at a native oyster on our coast. And she showed basically that if at some time in its life, this oyster experienced low pH or less than happy conditions, oystermen call it sweet water. So if they didn't have sweet water, they were forever smaller, no matter how good it got down the line. So they were canalized. They were forever changed by that exposure. And when we first started thinking about this, it just highlights the vulnerability of early stages. But it tells you another thing, and that is that natural variation in the environment really matters to that organism. And if you look for it in a different way, it can be incredibly important in how you think about ocean acidification and climate change issues. So I want to bring to you a study that just published recently by some fish biologists here on the East Coast. This is a tidal flat. You probably recognize it on the East Coast near Long Island. And these guys are out seining for this little guy, the Atlantic Silverside. And Hannes Baumann did this study, I think from the University of Connecticut now. And what they did was really clever. They knew they had this little fish that's in an estuary where the pH is changing, and they were measuring it. And this thing spawns from like April through July. And what they did was they just went out and collected throughout the season. And they correlated the babies that they raised based upon this. So these are pH data from that area they were collecting, where over the season, I think the middle line is the average, the top line is the max, and there's the min. So by the time you get to July, the pH is drifting down. And what they found was that fish that were spawned by their parents later in the summer did better in the face of low pH. And so their hypothesis is, and the data are wonderful, they're saying, look, you know, if there's natural variation in the environment, the parents are responding to it either by provisioning their eggs, the females are provisioning the eggs in some way, or there's some other, it's called epigenetic plasticity in these fish that's allowing them to withstand these conditions. But things like this just make you really turn and go, you know, there is plasticity in natural populations. We need to look for it, and we probably need to protect it in some way. So that leads me to what what might we be able to do? Because honestly, serving on science boards in California and other groups, you really get to this point where what are we going to do? Because the mitigation for low pH is really almost impossible. I didn't say this. Somebody once said, there's even all the calcium carbonate in the cliffs of Dover would not solve our problems in terms of buffering the ocean. And you don't want to make fun of it because it is a mitigation solution that could work locally, but it's beyond the reach of what we can do. So a lot of you probably have been thinking about this, but there are many people around the world and on this coast and my coast who have grasped this natural variation phenomena and realized that there are places on our coast like eelgrass beds and kelp forests in my neck of the woods, but here on the East Coast, eelgrass beds for sure. These places have a lot of photosynthetic plants, big beds of them. And we started wondering, you know, that winner and losers thing, how is that playing out in the environment? So one of my intrepid grad students, who, and I forgot to put a picture of her up here, took some of our sensors and put them out at these 2 island locations in the Channel Islands off our coast. And the purple line, if I get that right, the purple line comes from a site where there's a huge surfgrass bed, eelgrass bed. And the other one is just kind of more of a coastal site. So right away, it's the same ecotype. There's nothing different. All the invertebrates and the fish are the same. But the seagrass bed so greatly buffers and changes the pH of the water around our sensor that we were kind of stunned when we saw these data. Like, seriously? And so people all over the place are realizing this, so much so that this is a major argument for the restoration of these beds of surfgrass and eelgrass where they've been lost, in terms of, you know, preserving really good water for nurseries of fish and invertebrates. And so in California, we're actively looking right now within our marine protected areas because on our coast they were set aside to protect fisheries, but now we're looking at them and going, oh, savings accounts for the future. Possibly need to identify places where there are kelp forests and these big beds of macrophytes that are buffering ocean pH change. So that's my big argument, is that we went all the way from we're toast, the open ocean is acidifying, to wow, there's an incredible amount of variation on a small scale, and that's the sentence I want to leave you with is there's natural variation that's occurring on scales that are relevant to management so that agencies, game and fish, local communities can do restoration activities on this scale that will buffer future chemistry changes for their local waters. And that's something that I never would have been able to say a few years ago. So for the journalists, this is something that was shared to me by people who think about science in a sort of an interesting way. And that is, when you first start studying something and the science starts to come out, and this is what OA did, oh my gosh, confidence high, simple and clear, oceans acidified, bad news, right? And then we started doing more work and people get down in the trough, more nuanced and conflicting results are coming out. And it gets a little confusing because you'll talk to scientists and they're like, well, this is happening and that is happening. And that's actually how progress occurs, is that you have to get past the simplified state, get into the complexity. And now in the OA field, we're digging our way out. We're realizing that not all ecosystems are the same, that some organisms have the capacity to adapt and have plasticity, and furthermore, that we can act locally to deal with some of these things, right? So my message is more environmental data, act locally, work collaboratively because this stuff is expensive. No one group can do it by themselves, and don't give up because most of the time you're just like, oh my God, this is bad. But restoration is possible. This is not the end of it. Anybody under 25 in here, get to work, right? Thank you very much.